Chapter 10 Testing Differences. Inferential Statistics. Inferential statistics are used to draw conclusions about a population by examining the sample. They help researchers test hypotheses and answer research questions and derive meaning from their results. Inferential statistics use the theory of probability to investigate whether patterns found in the sample of study can be generalized to the wider population where the sample comes from. Inferential statistics aim to test hypotheses and explore relationships between variables and can be used to make predictions about the population. Inferential statistics are used to draw conclusions and inferences, that is, to make valid generalizations from samples. This chart shows you the differences between descriptive and inferential statistics. Descriptive statistics organize, summarize, simplify, and describe and present data. Inferential statistics generalize from samples to populations, hypothesis testing, and make predictions. The three assumptions of inferential statistics are: populations from which samples are drawn are normally distributed on the dependent variable. Participants are randomly sampled from a larger population, and participants are randomly assigned to categories or groups of the independent variable. Alternative and null hypotheses. Inferential statistics test the likelihood that the alternative or research hypothesis is true and the normal hypothesis is not. In testing differences, the alternative or research hypothesis would predict that differences would be found, whereas the null hypothesis would predict no differences. Degrees of freedom. Degrees of freedom are the way in which the scientific tradition accounts for variation due to error. It is represented by the abbreviation DF, and it specifies how many values vary in a statistical test. 4 analytical steps. First, the statistical test is applied to determine whether differences or relationships exist. Relationships are covered further in Chapter 11. The second analytic step characterizes the type of difference or relationship. Sometimes the difference that appears in the data may not be the difference the researcher predicted. The third analytical step assesses the statistical importance of the difference or relationship, knowing that a difference exists and whether it is the difference that is expected is not enough. A researcher must go one step further. Using the significance level, usually set at 0.05, a researcher cannot determine if the size of differences he or she finds can be accepted as real or not random. Alternatively, if the probability level is not achieved, then any differences found are not statistically significant and no conclusion can be drawn. The fourth and final step is where researchers want to move beyond the data tested and into the future. Thus, the fourth analytical step evaluates the importance of the observed differences or relationships and then generalizes the findings. To make this evaluation, researchers must assess all the steps in the research process. These steps include: how is the sample obtained? Were participants randomly selected? Were participants randomly assigned to treatment or control groups? To what extent does the sample represent the population in which the researchers are interested? Was the research design appropriate for the hypothesis and research question? Were the data collected reliable and in valid ways? Chi-square. Researchers choose chi-square as a statistical test when they want to determine whether differences among categories are statistically significant. Thus, a chi-square is the appropriate test when data for one or more variables are nominal or are categorical. The researchers compare the observed frequency, or the number of times the category actually appears, with the expected frequency, or the number of times the category was expected to appear. If the observed frequency exactly matches the expected frequency, then chi-square is 0. The greater the difference between the observed and expected frequency distributions, the larger the value of chi-square. And the larger the value of chi-square, the more likely that the difference will be statistically significant. One-dimensional chi-square. A one-dimensional chi-square is the statistical test for determining if differences in how the cases are distributed across the categories of one categorical or nominal variable are sufficient. Contingency analysis. The chi-square can also be used for examining the association between two nominal variables. In this case, participants are classified into two variables in relationship to each other. The statistic is also known as a contingency analysis or a two-way chi-square. The two nominal variables are arranged in a table where rows represent one nominal variable and columns represent a second nominal variable. The display of frequency data in this type of table is called a contingency table. Limitations of chi-square. Besides being limited to the variables of nominal data, the chi-square has other limitations. One limitation is that the chi-square is a test of frequencies. As a result, when the observed frequency is 0 in any cell, or when the expected frequency is less than 5 in any cell, the test may not be accurate. The second limitation addresses a number of variables the chi-square can test. Finally, the primary design limitation is that the test cannot directly determine causal relationships. The t-test. The t-test is used to test hypotheses that expects to find differences between two groupings of the independent variable on a continuous level dependent variable. First, participants or elements are categorized according to one of two levels of the independent variable. Second, the dependent variable is measured for all participants. Then, the independent variable scores of one group are compared to the dependent variable scores from the other group. The t-test is used to test the significance of difference between the names of two populations based upon the means and distributions of two samples. However, the t-test is also regularly used in quasi-experimental and descriptive research designs. Common forms of t-test. The first common form investigates differences between the means of two independent samples. This type of t-test is commonly used because researchers want to compare the mean scores of the dependent variable for two different groups of people. For the independent sample t-test, degrees of freedom are calculated according to the sample size for each of two categories of the independent variable. A second type of t-test is now known as a paired comparison because it compares two paired or matched scores. In this case, scores on the dependent variable are not independent. Two-tailed t-test. A two-tailed t-test is when a researcher asks a research question or states a hypothesis for which a difference in either direction is acceptable. The t-test is called two-tailed test. Researchers use the two-tailed t-test when they present a difference exists, but they cannot predict the direction of the difference. One-tailed t-test. Alternatively, a one-tailed t-test is when there is a specific difference identified in the research question or hypothesis. Limitations of the t-test. The primary limitation of the t-test is that it is designed to examine the differences on one dependent variable according to two groupings or categories of the independent variable. Thus, researchers cannot use the t-test to examine more complex communication phenomena. Analysis of variance. Analysis of variance is generally referred to as the acronym ANOVA. The statistical test compares the influence of two or more groups on the dependent variable and is represented by the symbol F. Variance is the dispersion of the distribution of scores on the dependent variable. The greater the variance, the more scores deviate from the mean. ANOVA extends the t-test in two ways. First, it can accommodate more than two categories of the independent variable. Second, the test can accommodate more than one independent variable. Thus, the difference between the t-test and ANOVA is the number of categories of the independent variable that can be tested and the number of independent variables that can be tested. ANOVA basics. Because it can deal with greater complexity in testing for differences, several other basic issues must be addressed. These issues include planned or unplanned comparisons, and between groups and within groups variance. These comparisons can be planned or unplanned. Planned comparisons are the comparisons among groups that are indicated in the research hypothesis. These comparisons are specified before the data are gathered. Unplanned comparisons are conducted after the researcher finds that a significant ANOVA exists. Unplanned comparisons are not predicted by the research hypothesis. Although ANOVAs are commonly referred to as looking for differences between groups of the independent variable, ANOVA actually tests differences between and among means. When differences are found, between groups variance exists. The groups vary enough to distinguish themselves from one another. To be significant, the between groups variance must be greater than the within groups variance, or the variation among individuals within any category or level. One-way and two-way ANOVA. One-way ANOVA tests for significant differences in the dependent variable based on categorical differences on one independent variable. For this test, one independent variable is measured at a nominal or a categorical level. There must be at least two categories for the independent variable, but there can be many more. In a two-way ANOVA, there are two categorical independent variables and one continuous level dependent variable. In testing the effects or influence of two independent variables, ANOVA can't determine the relative contributions of each independent variable to the distribution of the dependent variable. Additionally, ANOVA can test for the interaction effect or the combined influences that can occur from both independent variables simultaneously. Factorial ANOVA. ANOVA is not limited to a two-way design. Rather, it can also be used for more complicated factorial designs with 3 or even 4 independent variables. At a minimum, a factorial design would include at least 2 categorical independent variables, each with at least 2 levels. Often, factorial designs have more than 2 independent variables. This complex ANOVA addresses another issue, that is, in complex designs with multiple independent variables of nominal data, researchers will often choose regression over ANOVA as a statistical test. Limitations of ANOVA. The obvious limitation is that it is restricted to testing independent variables made up of the nominal or categorical data. The other limitation is really one of complexity. When researchers used 3 or 4 independent variables, differences found can be significant but confusing and difficult to interpret. This chart can help you determine the appropriate test of difference by identifying the number and types of variables in the research questions or hypotheses.